Through the 1900s, at least four hypotheses were offered to account for the evolution of ever more extreme female preference when it comes to mate choice. And after much heated debate, scientists have reached a consensus for what the right answer is. And it is, just kidding. There is no real final answer, at least not yet. Here's another one of those instances in which understanding the question is what's important. Why is this an interesting puzzle? The answer? Meh. It would be nice to have one. And if we had more time, we could spend another half hour going through the candidate explanations and still not come to a definitive answer. We don't have that time. But if you want a nice summary of sexual selection, I recommend this page from Nature Publications. I'll also put the link in Canvas so you could link to it directly instead of having to copy this down. Nature is the UK equivalent of the magazine Science, which is published by the American Association for the Advancement of Science, or AAAS. These two, Nature and Science, are weekly magazines covering the breadth of scientific fields, publishing the freshest and some of the most impactful research being done in the world. These are the two publications with the shortest turnaround time between submission of a manuscript, peer review, editing, and publication. If you want to keep your finger on the pulse of the world of science, I recommend that you check out their newsfeed, or even easier, subscribe to the weekly podcasts. But now it's time to close the book on microevolution and move on to the next section of our survey of evolutionary mechanism and process. Speciation. Okay, there's this thing we'll be discussing at length in BIO202. It's the biological species concept, which defines a species as a closed gene pool. Basically, draw a line around all the other organisms on the planet with which you could exchange genes, and that would be your species. An official definition would go something like this. If two populations A and B were brought together into the same general location in nature, here we'll use the term sympatry. Syn for together, patria for homeland. And while they're in the sympatry, they remain as separate gene pools, with A genes being kept pure because A's have babies only with A's, B's have babies only with B's. If this happens, then A and B would be distinct biological species. If, however, the A and B were to hybridize, and those hybrids made it with the A's, carrying the B genes in the A population, and maybe they also made it with the B's, carrying the A genes of the B population, then this could result in a melding together of A and B into a single mishmash gene pool. And this is the result that would occur, indicating that A and B are the same biological species. Basically, if A and B interbreed, they would not be different biological species. They only qualify as different species if they don't interbreed and maintain the isolation of their gene pools when they're there under natural conditions and in the same physical location in sympatry. This biological species concept will be our working definition for species, for a bit anyways. There are actually a few problems with this view on species, but we must start here because it gives us the best foundation to discuss speciation. The biological species definition is convenient more than anything because it gives us unequivocal clarity on what a species is, and in this way it also gives us an objective way of defining when a new species has formed. In other words, when speciation has occurred. How do new species form? Well, thanks to Ernst Mayr, we mentioned him in an earlier lecture. The gold standard answer to how species form is through the process of allopatric speciation. Allopatry, from allo for different and patria for homeland, means different geographical locations, and this is crucial for this kind of speciation. The process of allopatric speciation begins with two factions that start off as two populations of the same species that become geographically separated, each living in isolation from the other. Maybe a mountain range like the Rockies comes up, separating a western population from an eastern population. Or maybe it's a land-dwelling species that establishes populations on two distant islands. As a result of this isolation, gene flow between the populations is cut off to zero or near zero. The only other requirement for allopatric speciation is time. This period of isolation must be long enough to allow the two factions to evolve independently of one another, eventually reaching the point where even if they were brought back into sympatry, they would not be able to exchange genes. Let's draw a diagram. Here's an ancestral species A. It's successful in colonizing new habitats, maybe large islands out in the ocean, the Bahamas. The ancestral species is on the mainland in Florida, and it sends out migrants over the waves to establish populations in New Providence and Grand Bahamas. Now let's just call these populations 1 and 2. Initially, the island populations will be just like each other and like the mainland population from which they derived. But over time, each of the two island populations evolves along its own independent trajectory as a result of mutation. A mutation occurring on island 1 won't be happening on island 2, and vice versa. As a result of genetic drift, the population on island 1 becomes fixed for the big A allele, while the population on island 2 becomes fixed for little a, and also possibly as a result of differing natural selection. Maybe there are lots of bugs to eat on island 1, but none on island 2. But island 2 has an abundance on fruit, which are largely absent on island 1. You get the idea. There are the normal microevolutionary forces at work in both populations, all three of the populations if you count the mainland source population. But it's like each of the populations is doing its own thing, following its own path. Now, even after many generations of having these distinct histories of microevolutionary changes, the populations on islands 1 and 2 might still be able to exchange genes if they were brought into sympatry. Maybe after 600 generations, they're still likely to exchange genes. But if you allow the isolation on the different islands to persist for longer, you will eventually reach the point, maybe after 800 generations, when these two populations would no longer exchange genes if brought back into sympatry. And this would define the two populations as now being different biological species. This also defines an event of allopatric speciation. What was once one species is now two, and they diverged through a period of geographical isolation. One reason this model for the origin of new species is so powerful is because of its simplicity. Remember the principle of parsimony? Allopatric speciation requires only two conditions, both ridiculously easy to obtain: geographic isolation and time. That's all. Local populations get cut off from the rest of the species. This sort of thing happens all the time. How did the mainland finches get to the Galapagos in the first place? Finches are terrible at long-distance flights. They've got this characteristic flight pattern where they beat their wings and pause, fly and dip, fly and dip. If you ever see this, it's probably a finch. But this is not the energy-efficient style that you would need as a long-distance flyer. It might have been that a storm blew a whole lot of birds way out to sea, and only a few of these weak flyers made it to the safety of an island by sheer luck. Once established on the Galapagos island chain, the birds could theoretically fly from one island to the next, though this would have been a risky proposition, and so it wasn't done much, at least not deliberately. But now the finches could persist as an array of island birds, basically on their way to becoming different biological species. Land animals living isolated on oceanic islands is the classic case, but geographic isolation can occur on the mainland as well. Take the squirrels on either side of the Grand Canyon. I'm pretty sure it's in your textbook, but you can also Google Grand Canyon, squirrels, allopatric speciation. Allopatric speciation is also a powerful model because it could explain the majority of all speciation events that have ever taken place in the history of life. Not only is it parsimonious, having few requisite conditions, it also has some substantial supporting evidence from nature. This may sound odd to you because it's obviously not the kind of evolutionary process that you can recreate and study in the laboratory. Someone might sneer, but we've never seen speciation take place, so where's the proof? Well, we don't ever really prove our hypotheses. Remember the indirect nature of science? We talked about this in the last lecture. But in the case of allopatric speciation, it does make an important prediction that we can validate. If allopatric speciation is a thing, then we should expect lots of speciation in those situations where you have the kind of geography that's conducive to long-term isolation of populations. That is, islands. Our supporting evidence for allopatric speciation takes the form of large assemblages of relatively closely related species on island archipelagos that split away from a common ancestor relatively recently. And we have a small mountain of examples illustrating this. On the Galapagos, you have not only the finches, but you also have the tortoises and the iguanas. In the Caribbean, you have anolis lizards, as well as tiny fishes that live in freshwater streams. In the Philippines, you have gecko lizards, as well as shrews. On the Hawaiian Islands, you have the honey creeper and many other birds, as well as plants and insects. Of special interest are the Hawaiian fruit flies of the genus Drosophila. That's the same genus as the Drosophila melanogaster fruit flies of classic genetic research. In all of these cases, it would appear that speciation has been turbocharged by the special geography of island archipelagos, lots and lots of isolated populations. Archipelagos like these represent only a tiny fraction of the world's land surface area, and yet they have a disproportionately large number of species. So isn't this exactly what allopatric speciation predicts? The evidence supporting allopatric speciation as a mechanism is strong, and it's for this reason that this is our gold standard when it comes to mechanisms for speciation. Now, after that setup, answer this. Here are some facts. 71% of the Earth's surface area is covered by water, and roughly 70 of that 71% is saltwater, oceans, mostly. And of that 1% freshwater cover, most is in lakes and swamps, and only 2%, okay, that's 2% of the 1% of the Earth's surface is in rivers. Fish live in water. So let's start with this. Should most species of fishes on planet Earth be saltwater fishes or freshwater fishes? What do you think? Pause the video and write down your answer in your notes. All right. If you said saltwater because the vast majority of water on the earth is saltwater, and so the overall number of fish in saltwater must be greater, and so the species diversity will also be greater, well, you're technically right. There are more species of saltwater fish, and you're totally right about there being far more saltwater fish than freshwater fish in terms of biomass and number of individuals. But if you take our lesson on allopatric speciation to heart, you will realize that the oceans, while vast, are pretty lousy conditions for allopatric speciation. You don't have abundances of geographic barriers. Sure, the Atlantic Ocean is largely isolated from the Pacific, communicating only through the Arctic and Southern Oceans, and to a certain extent the Panama Canal. You can have coastal habitats at specific latitudes, isolated by great distance from other similar habitats. But you know where you do have a really large number of local populations isolated by geography? Rivers. So if you said freshwater, of course, because the fishes in one watershed or river system flowing into the ocean would be completely isolated from the fishes in other watersheds that flow into the ocean at different river mouths, congratulations. You're right about the mechanism part, which is the important part. And you're close to being right about there being more species in fresh water. The actual count is about 60% saltwater fishes and 40% freshwater. And here's the kicker. The majority of saltwater fish species live on reef systems. Now, if you remember anything from Finding Nemo, you'll know that for fishes living on and around reefs, the reef the fish calls home is separated from the next reef by a large distance of deep sea, in which there are lots of hostile and predatory baddies. And so, young reef fishes in Finding Nemo were taught never to stray from the home reef. Reef fishes also have another characteristic that's different from most saltwater fishes. Most saltwater fishes produce tiny, tiny eggs that are spewed into the water in mating swarms. The eggs develop quickly into tiny larvae that float around living as microscopic zooplankton until they're big enough to actively swim around with other tiny fishes in a school. But while they're planktonic, they have to go wherever the water goes. If reef fishes did this, then there would be no way to isolate the fishes onto one home reef. The zooplankton fish larvae would get scattered across the ocean by the strong currents. The chances that any would survive and actually make it to a suitable reef habitat would be really, really poor. And so it might not surprise you that reef fishes have evolved life histories that allow the young to be born large. Think of that opening scene in Finding Nemo, where Nemo's mom, rest in peace, laid large eggs, cementing them on the inside of a little crevice in the reef. Reef fishes like Nemo are born relatively large and already looking like a fish and able to swim and to learn to always stay close to the home reef. This life history character, large young, is important here because of the way it allows clownfish and surgeonfish like Dory and most of the residents of Marlin and Nemo's home reef to remain isolated from the other populations inhabiting nearby reefs. There are thousands of isolated reef habitats in the Great Barrier Reef and dozens of similar coral reef systems scattered around the world in tropical oceans. Reefs are biodiversity hotspots for fishes for the same reason that we have many species of Drosophila fruit flies in our 50th state. The same reason river fishes nearly match saltwater fishes in overall species number. And this is that allopatric speciation is valid as a mechanism of species proliferation. I'm about out of time with today's lecture, but there are a few more points that I need to bring up about speciation generally. In allopatric speciation, one ancestral species splits into two distinct gene pools, two separate species. Could there be a three-way split? Sure, in theory. But consider that the evolution of species defining differences between two isolates, the actual speciation event, happens randomly and it takes a long time. And it's only meaningful to us if both of the resulting species are successful, giving rise to its own lineage with a history that's distinct from the other. Now, of all the new species that ever form through allopatric speciation, how many of them are successful for a long enough time to even leave some fossil evidence? Probably not very many. Even if it does happen on occasion that one species splits three ways, for us to have a record of this would require that all three are successful, and this can't be a common occurrence. In our class, we will generally presume dichotomous, that is, two-way splits. Speciation events are demarcated for us in the history of life as a split in the phylogenetic tree. One gene pool, one species is there, and then poof, two gene pools. That poof is a speciation event. Every time the tree splits, that's a poof, an occasion in which one ancestral species gives rise to two derived species. We use trees extensively in BIO202, and it's important for you to understand the connection between what we talked about today and these nodes in the tree of life. Each split, it's a speciation event, and very likely an allopatric speciation event. This way of thinking about speciation with one lineage splitting into two is called cladogenic. Cladogenesis is also known as branching evolution. The word comes from clade, family. Think of the word clan, and genesis, origin. Cladogenesis is the process by which families of many related species descend from a common ancestral species. Allopatric speciation is a very important, likely the most important mechanism by which cladogenesis happens. The other way of thinking about speciation is where one species gives rise to a different species without a split. There's one species, one gene pool, at both the beginning and at the end of the process. This is anagenesis, where one ancestral species A is subject to microevolutionary forces for many, many generations, and eventually becomes the derived species D, which is declared as distinct from species A. Can this happen? Sure, it happens all the time. But this is different from cladogenesis in some important ways. For one, anagenesis does not account for changes in species number. When we take the big picture look at the history of life, one important dimension of evolution is change in overall diversity. The total inventory of species, genera, family, classes, etc. has been subject to increases and decreases, sometimes catastrophic decreases in mass extinctions. Cladogenesis is needed in order to account for the increases in biodiversity. We'll return to this in the last lecture of this unit. The other thing about anagenesis is that the two species, A and D, ancestral and derived, cannot be distinguished based on the biological species definition. You can't demonstrate that the species D that you have today would be unable to exchange genes with the ancestral species that was around 10,000 years ago. If you're going to declare A and D as different species, you need to have some different criterion for defining species, definitely not the biological species concept. Now, biological species is sound enough as a concept. After all, it allows us to define a speciation event under cladogenesis, but it's unusable in most situations. How often do we actually test the hypothesis that A and B are different species by putting them together in a sympatry and seeing whether or not they can exchange genes? Still, for a professional taxonomist, there needs to be a practical method for determining when we're talking about different species versus when we're talking about different populations of the same species. Defining the boundary around a species is not just an inconsequential exercise interesting only to museum curators. I'll give you an example. Historically, the four populations of wild giraffes in Africa had been considered distinct as subspecies, but they were all classified in the same species. Now, a couple of years ago, someone made a strong case that these are actually different enough to qualify as four distinct species, and this had large ramifications from the standpoint of conservation biology. As a single species with four populations, giraffes were numerous enough not to have any super protected status. But if they're split into four distinct species, two of these species would be small enough to qualify as endangered. And this could have significant impacts, for example, on the countries in which these giraffes range. Now, I'm not going to ask you to learn about the different methods used to define species other than the biological species definition. This is an executive decision that I'm making for BIO202 students in my class. I do want you to fully understand the biological species concept and to appreciate how it's the foundation for cladogenic speciation events where one lineage splits into two. We'll continue with this idea at the beginning of the next lecture.